Okay, so we have been discussing different receptors on macrophages, okay? And we previously discussed different types of scavenger receptors, you know? The macrophages are there in the tissues, they reside in the tissues, and they are there for longer periods of time. They are always vigilant. Whenever there is entry of some microbe into the tissues, these macrophages are there to just, grapple them or to just, respond to that kind of pathogenic attack. So the scavenger receptors are used for the purpose of phagocytosis in order to engulf that pathogen, degrade it with the help of different enzymes that are there in the macrophages, and get rid of the pathogens. Now, these are other types of receptors which are called the signaling receptors, and we also know them by different names, just like the Toll-like receptors or TLRs. You can also call them CD281 or up to CD290. These are 10 different types of Toll-like receptors, TLR1 to 10. Or you can call them CD281 to TLR1, and TLR10 would be CD290. Okay, so it's a family of these, different genes, 10 different genes that are coding different types of receptors, proteins. And these receptors, they instruct macrophages to recruit additional cells of the innate immunity to the infected tissue. And these Toll-like receptors are present on many different types of immune cells, but on macrophages, particularly for the detection of LPS or the lipopolysaccharides, which is part of the microbes or the walls of microbes which have lipopolysaccharides, macrophages have TLR4 or Toll-like receptor 4, which can bind with LPS on the microbial surfaces. These receptors, as you see here in the yellow, these have an extracellular domain that you see here, and they have a cytoplasmic domain that you see here on the inner side. The extracellular domain is also called the pathogen recognition domain, because this domain would be used to bind with the pathogen, okay? And the intracellular or cytoplasmic domain is called the TIR domain. And this domain is for intracellular signaling. When the pathogen is recognized here at the surface of the macrophage with the help of these receptors, then the signal is transmitted inside with the help of this domain of the Toll-like receptor. The signal would be transmitted inside to some other proteins. There are some adapter proteins that would be activated, and at the end the message will go in the form of a transcription factor, which will be released as a result of that cascade of events that is happening there. And that transcription factor would enter the nucleus and start transcription of different genes. That is what this process of recognition of the pathogen is leading towards the synthesis of different molecules like different cytokines or other molecules needed for the inflammatory response. Okay? This is the job of the macrophage, to initiate this process of inflammatory response. And for that purpose, it needs different types of molecules to be synthesized by this particular cell. So the Toll-like receptors are doing that particular job with the help of these different domains. The pathogen recognition domain actually is a 20 to 29 amino acid variable domain. There is a lot of variation in these amino acids. So there are many different types of these receptors that you would find on the surface of immune cells. And this domain is rich in hydrophobic leucine. There is a region which is known as the leucine-rich repeat region. This LLR determines the specificity, that leucine amino acid which is there is a leucine repeat region, and if you look at the variation in these receptors, the number of these repeats actually determines the variability there in receptors. And their internal domain, the TIR domain here, this is the tall interleukin receptor, or TIR. It is on the cytoplasmic side of the membrane, and this is named so because of this observation. This is a conserved domain as compared to the outer domain. The outer domain is variable, and it should be variable because there are different types of pathogens to be recognized by the Toll-like receptors on the surface of the macrophage. But the signal which should be generated here with the help of the TIR domain should be, a single one to lead to this process of inflammation, or to lead to a particular pathway that leads to starting this process of inflammation. So this part is conserved, the outer part is variable. This does not need variability, here, because it can just start with this process of phosphorylation and dephosphorylation of different proteins which are present in the cytoplasm and lead to, the release of transcription factors. And I told you that why it is called TIR, because this was first observed in the case of interleukin-1 beta receptors, which is a very powerful inflammatory cytokine. You can say that inflammation actually begins here. When interleukin-1 beta, it would bind with its receptor, interleukin-1 beta receptor. Okay? So as it was observed first in the case, this domain was observed to be part of the interleukin-1 beta receptor. That's why we call it the TIR domain. So this is the signaling domain. Now, if you see here, somebody is infected with a gram-negative bacteria which is present there in the tissues, okay? So what would the macrophage do with this gram-negative bacterium that you see here? The macrophage is going to engulf this gram-negative bacterium, the pink one that you see here. And what is the purpose of taking this gram-negative bacteria inside? The purpose is to degrade it, okay, and release different constituents of this particular bacterium, which would include the lipopolysaccharides as well. Lipopolysaccharide is part of the wall of gram-negative bacteria. It's most abundant there, okay? When the bacterium would be engulfed here, now it's here in the endosome. Okay, this particular peptide that you see here, LBP, this is a soluble, lipopolysaccharide binding protein, LBP. Okay, this is present there in the tissues normally. When it comes in contact with, some bacterial parts, it can bind with them. When the bacterium is engulfed here, along with the LBP protein or the lipopolysaccharide binding protein, now it's inside the endosome here. And these endosomal vesicles deliver the LPS to CD14 in exocytic vesicles. These are different vesicles that also contain this another, protein which is known as CD14. Now the bacterium has been engulfed. Lipopolysaccharide here is bound to LBP. The LBP is going to shift this lipopolysaccharide to CD14, and when it shifts this LPS, LPS binds to CD14, because it has a binding site for CD14. Now this endosome comes here at the surface of the macrophage or the membrane of the macrophage, and here the CD14 is just expressing this particular LPS which was bound here on its surface. What would happen next? Now MD2 protein, this is another protein which is going to recognize this complex, and there would be this formation of a complex of the TLR4, LPS, and MD2 on the surface of the macrophage. Okay, so what happened here? The bacterium was engulfed with the help of scavenger or phagocytic receptors by the macrophage here. What are those receptors? Our previous lecture, okay? Just like the mannose receptors that you see, Dectin receptors that we talked about, or Macroreceptors that we were talking about, or IC3B receptor CR3 and CR4. These are different receptors that we already have talked about in the previous lectures. All those are scavenger receptors, which are used for phagocytosis or increasing this process of phagocytosis to take that pathogen inside its body, So here the bacterium, when it enters the tissue, has been taken up with the help of all those receptors. Now LPS as it is being degraded, LPS has been separated, and LPS has been, shifted to another molecule there inside the macrophage, which is CD14. CD14 just comes here at the surface. It is now bound to LPS. This is recognized by MD2. MD2 has a binding site in this particular Toll-likereceptor 4, which is present here on the surface of the macrophage. This horseshoe-like receptors, TLR4, is for recognition of the LPS or lipopolysaccharides of gram-negative bacteria.